Welcome to Biochemistry Simplified. My name is Dr. ELIC, and we are here to do another exciting topic um, in biochemistry known as your biotechnology and genetic engineering. Today, we'll be looking at biotechnology and the genome. Okay, so from time immemorial, biotechnology has come to play. Man has been manipulating living things to solve problems and improve his way of living for millennia now. More than a decade, biotechnology has been practiced. Okay, so what then is biotechnology? So biotechnology can be described as a range of scientific techniques. And these scientific techniques includes genetic engineering, includes genetic engineering, that are used to create, improve, or modify plants, animals, and microorganisms. For just one purpose, for the benefits of humans. So that's what biotechnology is about. It actually has a end goal for benefiting humans, and they do that by applying a range of scientific techniques, um, which includes genetic engineering to be specific, that is used to create, improve, modify animals and plants, and even microorganisms for the benefit of what humans. Biotechnology, its brief history. So we want to look at the brief history of biotechnology. Like I said, it's been in existence for long, in millennia now. So let's quickly look at the brief history. Let me do this in red. The brief history of biotechnology. Okay, so biotechnology at the beginning of the 20th century began to bring industries and agriculture together. They began to bring industries and agriculture together when? At the beginning of the 20th century. So the biotechnology starts way back to the beginning of what? The 20th century. So we say biotechnology at the beginning of the 20th century began So it actually started at the beginning of the 20th century. It began to bring industry and agriculture together. So it started as far back as that. Okay. Point number two is brief history stating the application of biotechnology. So during World War One, fermentation processes were developed that produced what acetone from starch and paint solvents for the rapidly growing automotive industry. So it started at the beginning of what 20th century right here, and here you have during the World War I, fermentation processes were developed fermentation processes were developed that produced acetone from starch and paint solvents the rapidly growing. Okay, so that's point number two, talking about the brief history. All right, so still on the brief history, we look at World War II, what happened in World War II as it relates to biotechnology. So World War II brought World War II brought the manufacture of penicillin and biotechnology. And biotechnological focus moved okay, moved in pharmaceuticals. So the Cold War years were dominated by work with microorganisms in preparation for biological warfare, as well as antibiotics and fermentation process. So the manufacture of penicillin and biotechnology focus moved in excuse me moved in pharmaceuticals. Thus, the Cold War years were dominated by work with microorganisms in preparation sorry as well as antibodies and fermentation processes. So it's great to know that biotechnology started in this pattern. Biotechnology is one of the powerful tools of the 21st century, which has actually revolutionized the life sciences and has grown as a separate discipline encompassing a number of disciplines like encompassing a number of disciplines like the cell biology, immunology, molecular biology, recombinant DNA, recombinant DNA, and bioinformatics or bioinformation. Okay, so biotechnology has emerged as such, right? So, still on the brief history from the 20th century going down towards the 21st century. So, still on that brief history, we say that biotechnology, now getting to the 21st century, we are saying that biotechnology of the powerful is one of the powerful technologies of the 21st century. It revolutionized the life sciences and has grown okay so and has grown as a separate so it has grown as a separate discipline. That's true. Biotechnology has grown as a separate discipline, which now encompasses okay which now encompasses encompassing a number of disciplines such as you have one cell biology, you have immunology, you have molecular biology, then you have recombinant DNA. Recombinant DNA. Recombinant DNA simply means recombinant deoxyribonucleic acid. So you have recombinant DNA for short, which simply means recombinant deoxyribonucleic acid. And you have bioinformation, of course, that includes bioinformatics, bioinformation, full stop. All right, so we have seen the brief history of biotechnology coming down all the way from the 20th century to the 21st century. And in seeing that, we have seen in the 21st century that it has become one of the powerful tools. Now, biotechnology is uh, currently being used in many areas such as agriculture, bioremediation, and food processing and energy production. All right, so yeah, it's currently being used in these areas, and it seems to be leading a sudden new biological renovation. Okay, so it seems to be leading a sudden new biological revolution, and has brought us to the brink of a world of engineered products that are based in the natural world rather than on chemicals and industrial processes. Okay, so coming down through the the history of biotechnology, currently we say, let me do this in another color because we have looked through the brief history from the 20th century to the 21st, and we are currently, so biotechnology is currently being used in many areas, which includes: one, we have agriculture; two, we have bioremediation; three, we have food processing; and four, we have energy production. So these are the basic areas of what um, areas that biotechnology has currently covered. Okay, so and it has brought us to a brick of a world of what engineered products. Okay, so all organisms, actually that is currently where we are at biotechnology. So we begin to now look at the systems of what biotechnology and what it's all about. So I'll do that on the next page. Okay, so um all organisms consist of small cells, typically too small to be seen. So how does biotechnology come in, and how is genetic engineering done? Okay, too small to be seen said all organisms consist of small cells, typically too small to be seen by the naked eyes, but big enough for optical microscopes. Each cell is a complex system consisting of many different building blocks enclosed in a membrane bag. So, describing the cell and how it's formed and its what location. Okay, so basically, biotechnology uses the cell of the of both unicellular and multicellular organisms. So we want to note that unicellular organisms or living organisms could either be what unicellular or multicellular. So living organisms either be unicellular, which consists of consisting only of one cell, consisting only of one cell, or living organisms could be what? Multicellular. Of course, your guess is as good as mine. Okay, so multicellular consisting of what? Many cells put together. Multicellular consisting of many cells. So bacteria and the baker's yeast are example. Okay, bacteria and the baker's yeast, which is your Saccharomyces cerevisiae, and the baker's yeast are examples of what? Unicellular organisms. Okay, examples of unicellular organisms. Why man why man and tilapia fish are examples of what? Multicellular organisms. So now what's the difference? So the difference between one organism and another are encoded by what? Genetic material. The difference between one organism and another is the encoding of what? The genetic material. So we are gradually talking of how these organisms which are divided, how living organisms which are divided into unicellular and multicellular are connected with what? Genetic engineering. Okay, so we say that the difference between one organism and another are encoded in the genetic material called what? The DNA, called the deoxyribonucleic acid, DNA. Now, DNA is the main information carrier molecule. DNA is the main information carrier molecule. And what does it do? Carrier molecule in a cell. So it carries information. So, and it has nucleotide bases that are grouped into what? Purines and what? Pyrimidines. So we just look at that. DNA, which is the genetic material for all living organisms, is what? Is the main information is the main carrier information carrier molecule, sorry, in a cell. Four different Four different nucleotides are grouped into two types. And you have the purines, which includes your adenine designated by A, and guanine designated by what? G. Then you also have includes what? Grouped into what? Pyrimidines. Purines and pyrimidines. And the pyrimidines consist of what? Cytosine designated by C, and what? Thymine designated by what? A T. Okay, so this forms the DNA molecule structure. These purines and pyrimidines forms the DNA molecule structure. Alright, so these purines and pyrimidines here, these forms it forms the DNA molecule structure. Great. So what does this do? Specific primers. Okay, what does this do? Let me show this in say red. So specific primers, pairs of nucleotides, can form weak bonds. So these purines and pyrimidines, they form weak bonds that their bases can bind to themselves. Okay, so these specific primers, oh, sorry about my pen that is acting up. Specific pairs, rather, not primers. Specific pairs of nucleotides can form weak bonds, such that your adenine can bind A can bind to T, which is your thymine, while C binds what? To G, which is your guanine. And this DNA molecule structure is shown below. So the DNA molecule structure is shown below. So let's see your 3' here. Okay, you have C, G, T, A, G, C, A, C, G, T, T, A, G, C. So, and that will give you your what? 5' N. Now, if you want to look at what we just said, they form weak bonds, okay, and as such, A can bind to T and C can bind to G. So this is C here, it will automatically bind to your G, and G will bind to C, okay, and T will bind to what? A will bind to T, C, G, okay, they are binded by what? Weak bonds. A, G, C. So AA binds to TT, TT binds to what? AA. and your A here will bind to what? T, and your G will bind to C, and your C what will bind to what? G. So that is what your 3' end to your what? 5' end. So this figure right here shows the base pairing of nucleotides in a DNA molecule. So we could label this figure as what? Figure 1. Figure 1, which shows what? The base pairing of nucleotides in a DNA molecule. In a DNA molecule. Okay, so this is basically that for the structure. You will notice that the end of the polynucleotide are marked with 5' and what? 3'. This has chemical reasons in the numbering of the OH group of the sugar what? Ring. So by convention, DNA is usually written with 5' to the left and 3' to what? The right. So if that is correct, this should be our 5' to the left and 3' towards the right, and automatically this will be your 3' here to the left and what? 5'. Alright, so it's usually right, left, and 3' right, with the coding strands at what? The top. So describing this structure here, we say that the end of the polynucleotide are marked either 5' or 3'. This himself has a chemical reasons in the numbering of the what? OH group. OH groups of the sugar ring. However, by convention, DNA is usually written the coding strands at the top. Okay, so that's how DNA is written. You have the coding strands, which is this right here. These are the coding strands. It's like a strand 1 at the top, and it's always written from 5' to 3'. Okay, so two of such strands are termed complementary, as A can mutually be exchanged with T and C with G, while changing the direction of the molecule to the opposite side. Okay, so two of more of this still on this figure here, the DNA molecule, base pairing of DNA molecule. We say that two note. So we say two of such strands above two of such strands above, or sorry, as shown above, as shown above, attempt complementary attempts complementary. Here you have A can which is mutually exchanged such that your A, which is your adenine, can mutually be exchanged with T, which is your thymine, as well as C, which is your cytosine, with G, which is guanine, and changing the direction of the molecule to the opposite. And changing the direction of the molecule to the opposite. Great. Okay, so when you change the direction of the molecule to the opposite, you have these strands of the DNA, as we saw earlier, that they are what's complementary to each other. We saw that they are complementary to each other, and apart from the basis, okay, apart from the basis, DNA molecule is the main. Okay, before that, by changing the direction of the molecules, now it is worthy of note that such interactions are individually weak when two longer complementary polynucleotide chain meet, they tend to what? Stick together. So regarding this strand right here, okay, it's important to note that although they are complementary, okay, although such interactions are what usually weak, when two longer complementary polynucleotide chains meet, they tend to what? Stick together. Okay, they tend to stick together. So I want to put that down here. Also note that also note that also note that such interactions are individually. What interactions now? This weak this weak bond between pairs that form weak bonds, whereby T is binding to what? A, and C is binding to what? G. So also know that such interactions are what? Individually weak. They are individually weak when two molecules, two longer when two longer complementary polynucleotide polynucleotide chains meet, what happen? They tend to what? Stick together. So as a result of the weak bonds, if you have longer polynucleotides, they tend to what? Stick together. Apart from that chain, there are other things other from the basis. Apart from the basis, which is the nucleotide basis, the DNA also have other things it is made up of, and we'll begin to look at those things subsequently. All right, on the next page, the those other things that DNA are made up of. So we say apart. Let me do this in another color. It's blue. Sorry about that. So we have apart from the basis. When I say basis, we are looking at those nucleotide bases, purines and pyrimidines, nucleotide bases, where you have adenine and guanine, and then cytosine and what? Um, Adenine and thymine and cytosine and guanine. So you have those nucleotide bases. Apart from them, we say that your DNA molecules are also made up of they are also made up of sugar phosphate, also made up of sugar phosphate backbone backbone. So this phosphate backbone is shown below. So as shown, they are made up of sugar phosphate backbone as shown below. So you have your nucleotides and your nucleotides. Apart from them, you will see the sugar phosphate backbone shortly. So you have this. Sorry about that. So you have so this is one nucleotide right here, and we have another one. Okay. Alright, so right here you also have your sugar moiety. So right here and this right here forms what? your sugar phosphate backbone. Sugar phosphate backbone. So from this structure, you see that nucleotides are the building blocks of what? Your DNA molecule. See that your nucleotides are the building blocks. They are the building blocks of your DNA molecule or of a DNA molecule. Great. So they are the building block of the DNA molecule, and they form the three. We begin to now look at the structure of this DNA molecule, specifically the three-dimensional structure. So we've seen the DNA, which is the genetic materials, and they are made up of nucleotides, which are said to be the building blocks of a DNA molecule. And apart from that, the bases that are seen, you have the sugar phosphate backbone as seen in the structure. In our next set of lectures, we'll begin to look at the three-dimensional structure of the DNA molecule.